Hello everyone, today we are going to study about PDH complex. In earlier video we have studied about glycolysis and product of glycolysis was pyruvate. Now in cellular respiration pyruvate has to enter into Krebs cycle. For this, pyruvate has to convert into acetyl-CoA. Acetyl-CoA. For this conversion, we need a group of enzyme, that is E1, E2 and E3. And this group is known as PDH complex, that is pyruvate dehydrogenase complex. Pyruvate dehydrogenase complex. We know that glycolysis takes place in cytosol and Krebs cycle takes place in mitochondria. So this pyruvate has to enter into mitochondria from cytosol. As we know the structure of mitochondria, mitochondria is a double membrane bound structure, outer membrane and inner membrane. Now outer membrane is permeable for pyruvate. So pyruvate will enter into outer membrane without any hurdle. Now pyruvate has to cross this inner membrane and inner membrane is impermeable to pyruvate. So there are present pyruvate transporters on this inner membrane and there will take place an electroneutral exchange. That means pyruvate which have negative charge will enter into mitochondrial matrix and OH negative ions will move toward intermembrane space. Now let's talk about the main reaction. In this reaction, pyruvate, which is 3-carbon compound, will convert into acetyl-CoA, that is acetyl-CoA. It is an reversible oxidative decarboxylation reaction. So this CO2 will release in this reaction. CO2 will release in this reaction and here one NADH is also produced. Operation of PDH enzymes and coenzymes will take place here. Now let's discuss about enzymes and coenzymes that participate in PDH complex. First one, we have 3 enzymes that are pyruvate dehydrogenase and we can denote it as E1 unit. Second one is dihydrolipoyl transacetylase, that is E2 subunit. Third one is dihydrolipoyl dehydrogenase, that is E3 unit. And all the 3 enzymes collectively known as PDH complex. Now coenzymes, first one is thymine pyrophosphate This is the coenzyme of pyruvate dehydrogenase enzyme. Second one is FAD and third one is NAD. Both participate in oxidation and reduction reactions. Next is coenzyme A. This will provide us acyl group for formation of acetyl-CoA. Last one is lipoate or lipoic acid. Lipoic acid consists thiol groups which can go reversible oxidation and form disulfide linkage or disulfide bonds, and this lipoic acid act as swinging arm. Let us discuss about E2 subunit because this is the largest subunit of whole complex. In this E2 subunit we have three domains. First domain is trans-acetyl domain. Second domain is binding domain. This domain is responsible for binding of E1 to E2 and E2 to E3 unit. So this is responsible for integration of the whole complex. Last one is lipoyl domain. And this domain acts as swinging arm. Let's understand how these reactions takes place in PDH complex. We have pyruvate here. Pyruvate will attach to thymine pyrophosphate and release CO2. This CO2 is released from the first carbon of pyruvate, and thymine pyrophosphate will be attached to the second carbon of pyruvate. And this will form hydroxy ethyl thymine pyrophosphate, and this reaction is catalyzed by pyruvate dehydrogenase enzyme. In next reaction, here 2 electrons will be released when thymine pyrophosphate will regenerate. This lipoyl moiety will interact with hydroxy ethyl thymine pyrophosphate, and electron released in this reaction will interact with this lipoyl arm and form acetate. This group will be attached to this sulfur group of lipoyl arm, and this will esterified to form this group. Now CoA will enter into this reaction, coenzyme A, and acetyl-CoA will be produced. And as such our lipoyl arm will again reduce to this form. Now when this form will convert into its original form, 2 electrons will be released in form of energy, and that was received by FAD, and FAD will be converted into FADH2. Now when FAD will regain its form, that is FAD, NAD will take its electron and convert it into NADH. As we have studied in the reaction, one NADH will be formed in this reaction. This step was catalyzed by dihydrolipoyl transacetylase enzyme, and this step is catalyzed by dihydrolipoyl dehydrogenase enzyme. Dehydrogenase enzyme. So first unit, second unit, third unit, and these are the enzyme. What is the special thing about PDH complex? That none of its intermediate will leave this complex, and this will enhance its efficiency. So in this reaction we have CO2 as a byproduct and NADH will be formed as energy, and acetyl CoA, this will further take part in Krebs cycle in mitochondria. Thank you so much.